Epibiosis is a biological association in which a living organism, known as epibiont, grows on another living organism's surface, called basibiont. Epibiosis is expected in the marine environment, where several macroalgae species function as basibionts, as they provide attachment surfaces to many epibionts. Among the macroalgae, brown algae of the genus Sargassum provides a habitat for many marine organisms, including invertebrates from the phylum Cnidaria. Among the cnidarians, polyps from the class Hydrozoa, generally known as hydroids, are common epibionts on both benthic and pelagic Sargassum species. Hydroids are successful epibionts because they can attach to artificial and natural surfaces employing their unique morphology, which, in a general way, consists of the hydrans, the gastrozoid polyp, the hydrocaulus, the stem of the colony or the individual, and the hydrorhiz, a net that allows the hydroid to attach to a substrate. For centuries, the knowledge and description of the Sargassum species involved in the epibiotic associations have been done in different areas of the globe. In this study, we aim to 1. Compile the worldwide knowledge of Sargassum species and their epibiont hydroids through a revision of ecologic, taxonomic, and faunistic studies. 2. Establish comparisons between the worldwide Sargassum species studied to date that are basibionts of hydroids. And 3. Contribute to the knowledge of the epibiotic hydroids of Sargassum in Mexico through recent reports obtained in La Paz Bay between 2018 and 2020. To fulfill our objectives, we search for Sargassum records in ecologic, taxonomic, and faunistic studies worldwide. We sought through the Google Scholar web search engine and include the words Sargassum and Hydrozoa. We extracted information from 128 peer-reviewed papers and theses and 4 studies where the author was indicated but the year was not specified, from 1816 to 2020. All the information was collected in a database that had the following elements: Sargassum and hydroid species taxonomic classification, reported and valid names, phylum, class, order, suborder, family, genus, and species, and geographic information as ecoregion, ocean, and country or sea. The number of Sargassum species that had been recorded as basibionts for hydroids around the world is 28 from the 361 species reported worldwide, which accounts for 7.7% of the total species of Sargassum described, and from which 26 are benthic species. The 2 species of holopelagic Sargassum, Sargassum natans and Sargassum fluitans, were also recorded in many analyzed studies. Nevertheless, in several studies, the Sargassum species is not specified. The most reported species throughout the analyzed literature was the holopelagic species Sargassum natans, being reported in 14 studies, followed by Sargassum zymosum, reported in 9, and Sargassum fluitans, reported in 8. Both of the holopelagic Sargassum species have been studied and reported as basibionts of hydroids around the world. Studies have reported Sargassum natans without detailing its morphotype, while 4 studies indicate one or more of its morphotypes, which are Sargassum natans 1, Sargassum natans 2, Sargassum natans 8, and Sargassum natans 9. Sargassum fluitans had been reported in 8 studies. 4 studies did not specify the Sargassum fluitans morphotype, while 4 have indicated one or more of the morphotypes 3, 5, and 10. Only 7% of the species have been studied for analysis of its hydroid epibiota in the benthic species. We found 125 species of hydroids as epibionts of Sargassum. Most hydroid species belong to the suborder Leptotecata, with a total of 101. The rest of the hydroid species, 24, belong to the suborder Anthoatecata. The Sargassum recorded species with the highest number of hydroid species was Sargassum cymosum, with 28, followed by Sargassum furcatum, with 22, and Sargassum polyceratium, with 18. This map portrayed the countries or seas of the world where studies about the Sargassum diversity of epibiont hydroids have been done. The highest number of studies have been carried out in the US, with 28, followed by Brazil with 16, and the Sargasso Sea with 13. There are 6 studies to date for Mexico, performed in the Caribbean, Gulf of Mexico, and the Gulf of California. In La Paz Bay, we found hydroids in the benthic species Sargassum lapacianum and Sargassum horridum, adding one new species to Sargassum species knowledge of basibiont hydroids, growing this knowledge in a 0.33% worldwide. In conclusion, only 7% of Sargassum benthic species are recorded as basibionts of hydroids, meaning that this association is understudied and has mainly focused on certain types of Sargassum, especially holopelagic. Also, Sargassum species studies are scattered worldwide, but some countries have several studies, while the majority do not have any. In this study, we add 0.33% to the knowledge of Sargassum benthic species and hydroids association through one new reported species, Sargassum lapacianum. The study of macroalgae basibiont species and their hydroid epibionts deserves a lot more interest to unravel this significant association.